Welcome to this session where we're going to talk about genetics and how genetics can help to also identify therapies. So I hope you will enjoy the next 30 minutes or so, and I'm very happy to answer any questions you may have in the end. So first, this is my disclosure slide. I'm involved in a number of things that could be seen as conflicts. And what I really want to talk about today is go a little bit about what we do currently in CMT genetics. In this time frame, really, that we have available here, there is, I can only show you sort of the big picture, but I do want to go into a recent success story, which is the SORD neuropathy success story, and you will see what that means. Okay, so as you know and have seen many times, CMT is a disorder that affects the nerves that run in your legs and in your arms, and it is important to sort of realize when you look at such a nerve, there are basically two kinds of CMT or peripheral neuropathy that are there. One is the CMT type 1, where really the nerves are surrounded by this insulating sheet of cells. It's almost like an electrical cable with the insulation. And those nerve the Schwann cells, they are sick in CMT type 1. So very different, quite different is type 2, where the nerve, or if you want, the electrical cable itself is damaged. So this would be type 2. For patients, often there is not really a difference. They can look quite similar, these disorders. We have also then, over the years, realized CMT is really much more than just nerves getting sick, peripheral nerves that run in your legs, say. We have found patients that also complain that their eyesight is affected, that their general brain function might be affected, their muscles might be affected, and so on. So the idea really is that what you experience as a CMT patient with the weakness and the sensory symptoms is really sort of kind of what is visible, but under it there could be other issues going on. For genetics, this is extremely important to understand, because you see here in these boxes all these cryptic names. These are all different genes, and we think that some of these genes are typical for certain combinations of symptoms. Here's another view of this, how these are many, or if not all, of the known CMT genes currently, and they sorted in different ways, really into dominant genes and into recessive genes. What this means is they are given down from the parents to their children in different ways. There are different ways of inheritance here. So dominant, typically one of your parents would be affected, but there could be exceptions. And in recessive genes, parents are typically not sick. They are carriers, but children might then have this CMT. So the point here really is, again, you see many different genes, over 100 genes now that we think are involved in, that can cause CMT. Now, to be clear, a single gene is enough to cause the CMT. And what this means is there are different types of CMT. There are over 100 different types of CMT out there. What this might mean for therapy, when you think about it, is that we might need dozens and dozens, if not 100 different types of therapy, which is a daunting concept, given that we have no therapy today. So, and this has always been an issue. Now, to discover all these genes, here's a sort of a time scale going back to almost over 100 years, really, from when the disease was first described in patients in the 19th century. And then only in the 1980s or so, geneticists started to understand which regions of the genome might be affected in patients with CMT. And then in the 1990s onward, scientists started to find individual genes. Again, you see these cryptic abbreviations here. These are the gene names, and you may notice here PMP22. Some of you might, this is CMT1A, for instance. And so these bars here really show you per year how many CMT genes were discovered. And the real idea of this slide is to show you how there are these waves of discovery, and we are really in the middle of another wave right now. And they are very much driven by technology. So the technology matters that's available, and it's always evolving, which is a good thing. Now, with all these successes and all these 100 different genes, the reality is today that an estimated 50% of CMT patients that show up in a clinic might not receive a diagnosis, even testing all these genes. So there is this big question mark: what other genes are out there? What are we missing? Okay, and CMT is not alone. This is a related disorder called hereditary spastic paraplegia. Very similar situation. They also have close to 100 genes in this disease, and many patients are not diagnosed. So that's just to show you that genetics is ever evolving. Despite spectacular success, we have a lot of work to do still. So one thing that has been a huge success in the CMT world of genetics is that we have managed to bring together a lot of genetic data from CMT patients within the United States mostly, but also within consortia we have, where, for instance, the United Kingdom plays an important role, but also Italy. But then also beyond that, really around the globe. So physicians who work on CMT, they have been able and willing to share genetic data they have from their patients anonymously with us. And we have created this database we call Genesis, which is an ever-expanding resource. And that database has really empowered a lot of the discoveries of these genes. So we currently, for instance, have close to 2,000 data sets from CMT patients. These data is available not just to my group in Miami, but really to any qualified CMT researcher out there. And that's really the point, to share genetic data more, discuss it internationally, and that really accelerates the discovery process. Also, there are other large collections of other diseases. Again, this HSP disease here, they are related in a way to CMT, and that can also sometimes help to discover new CMT genes. Okay. So here's sort of a brief peek behind the scenes. This is what researchers would see when they work with this database, and you know, the idea is really to make it not too cryptic, so that really all kinds of researchers can work with this, not just sort of computer-focused scientists, but also physicians or researchers who work more in a traditional laboratory environment. So this tool, this database, has really helped to bring everyone to the table and look for these genes. You probably heard a lot in other areas about machine learning and artificial intelligence. And yes, also in CMT genetics, we are using these tools now very actively. In fact, we have developed here in Miami this we call the Maverick Score. It's a neural network, we call this. This is basically a bit like, imagine a chess computer that predicts or that suggests the next move in a chess game. This score suggests which genes might be important in CMT to the scientist. So there's an interactive work here between the machine learning neural network and the scientist who is trying to understand CMT genetics. But we also use quite traditional methods, like something we call the distant cousin project here. Still, if there are large families of CMT that carry CMT in them, they in itself today can be a resource to identify a novel gene. So a large family, for instance, like this here, where despite everything, the CMT gene has not been identified yet, we can, with the technology we have available now, we have a good chance to actually find something. And distant cousin, why is it called like that? Because the further you go out from yourself in this family tree here, you know, to your mother, your grandmother, brother of the grandmother, and so on and so forth, the less you share with your ancestors, right? So you would share only 3% of your genes with this distant cousin. And if we would study this distant cousin and your genetics, we can really filter out just these, we can look just at these 3% of your genome instead of everything. So that's extremely powerful. If you're interested in this or think like, oh, this looks like my family, you can contact us, you can contact CMTA. They will definitely let us know, and we were happy to study this. This is another sort of typical result of these collaborative studies. A novel CMT gene we found a few years ago called ATP1A1. That, you know, was discovered in multiple families from across the world, and that was only possible because we have the data aggregated in this Genesis database in Miami. So when we found it, we had a family from Dr. Shai in Iowa. I work very close with Dr. Shai, but also from Dr. Seeman in Czech Republic. We had a family from southern Italy, from South Korea, and 2 families from different places in Australia. And here's just another example, a gene that was very recently discovered or published really, CAT-M3. And again, it was a collaboration between Miami, Dr. Shai, and the team around Mary Riley in London. And this is a kind of a rough overview that's probably not 100% up to date here. There are several new CMT genes per year really at this point, but all the bars that are on red, these are all the genes that were discovered by the INC or with the support of the INC and the Genesis database. So it's this idea of bringing data together is extremely important. Another sort of very interesting discovery was made 2 years ago and led by the group in London, Mary Riley, Henry Holden, Andrea Cortese. They discovered something very curious. Normally, all CMT genes carry mutations. I'm sure you have heard of mutations, where single changes in a gene causes CMT. Now, CMTA is a little different. There's a whole gene duplication that by itself is certainly also a bit special. And they found something else that is special. They found that there was a piece of the chromosome in these patients where the genome curiously is expanded. So there is more sequence there than there belongs there. And also that elongated piece of DNA was not in the gene itself. It didn't affect any of the protein-making pieces of the gene, but it was sort of in these regions that sometimes are referred to as junk DNA, like DNA that supposedly has no function. And so this expansion in these intronic regions, that is really the novelty here. And this disease has since intensely been studied. It was published on the cover of Nature Genetics. And it is actually quite common. It's recessive, and it often starts as a sensory neuropathy. So not so much a motor function involved, although it can be. Although it says here ataxia in this first paper, it's really to a large degree a sensory neuropathy. And these expansions could, for instance, explain more of these 50% of patients that we don't find genes today. So we have for a few years now worked a lot on finding more expansion examples for CMT. We haven't found new ones for CMT, but we have found new ones for other diseases. So, however, I think they are very difficult to study, and we might find more in the future for CMT as well. And now I want to come to this very exciting discovery, which was also published on the cover of Nature Genetics. So that's always a very high distinction for the significance of a scientific discovery. And what we found is there are mutations, traditional mutations in this gene called SORD, and it is a recessive type of CMT. Patients have more motor, they have more weakness, okay, than they have actually sensory problems, but they can also have sensory issues. So we identified this gene, and now what's so special about this one? You saw all the other names. Why is this so special now? It is special for a number of reasons. The main reason is possibly, again, a screenshot from the database that we found a lot of different families. A lot of families seem to have it when we found it, which made us think maybe this is not such a rare form of recessive CMT. Maybe this is more common. We also saw that all these patients here, they all share the very same mutation. And often CMT, that's not the case. Patients, they carry all kinds of mutations, not the same. So we then, looked at these families, and what we saw is that most of these patients were actually the only case in their family. The parents were healthy. They didn't have siblings that were affected, although some had. But many of these families you see here, only one person had it. So, and those are really the hardest cases, also for doctors to understand. If there's no family history, then it's not 100% clear if it is a genetic form of neuropathy. So solving a lot of these we call sporadic cases is extremely important. So very exciting here. These are as a brief overview. We found other mutations as well. So it's not just one, but this one here, most patients carried this particular change. And when we then look closer for this particular change, how common is this particular change in the general population? We found that about 1 in 200 people of all the people around you in different countries and different ancestries, 1 in 200 carries one of these changes. So now to get CMT, because it's recessive, you need 2. So with 1 copy, you're a healthy carrier. You wouldn't get it. So 1 in 200 are healthy carriers. That's a pretty high frequency. But you need 2 to be sick. So from this, though, we can calculate how many people are there in the United States alone that have 2 copies. Now, this is a statistical estimate, but I think there are over 3,000 individuals in the United States alone who carry 2 bad copies of this. Okay, so this makes it very likely the most common recessive form of CMT. Okay, this is sort of the CMT1A of recessive CMT. So what is this SORD thing, this SORD gene? It actually has been studied for decades because it was involved in diabetic research for a long time. And it is involved in this pathway, which is called the polyol pathway. Okay, so this is the polyol pathway. A little bit of biochemistry. Don't worry. This is how this works. There's glucose. You probably know what glucose is. This is the stuff that is high in people with diabetes. It's high in your blood. So you have too much, when you have diabetes, you have too much glucose in your blood. Now, because this is such a thing you don't want to have too high, there are different enzymes to keep it low. Insulin, insulin, possibly from diabetes, but there are other enzymes like this one called aldose reductase. If there's a lot of glucose, aldose reductase starts working. And what it does, it creates sorbitol out of glucose. It takes glucose and makes sorbitol. Sorbitol is something that is normally extremely low in your blood. You don't want it. So that's why immediately another enzyme, the sorbitol dehydrogenase, kicks in and turns the sorbitol into fructose. So this is fruit sugar, pretty harmless. And then you don't have sorbitol anymore. Okay, so that's the typical pathway. In these patients, the sorbitol dehydrogenase gene here, the SORD gene, does not work. So what happens if this doesn't work? The sorbitol goes up because that enzyme doesn't know that doesn't work. So it keeps making sorbitol. Sorbitol levels go up in the blood. And I'll show you, they go up a lot. And that is with great certainty the reason why people get the neuropathy. They have too much sorbitol in their blood. And there have been strategies developed actually over 20 years ago, how to correct high sorbitol in blood. And this was developed because of the diabetes connection. These drugs are called ARIs, or aldose reductase inhibitors. So they block this enzyme here. If this is blocked, then glucose is not made into sorbitol in the first place. That's the idea. Okay. So we have then looked at patients with these mutations, and what we see is that this is what we call a Western blot. Don't get scared. These are controls, and really what you have to focus on in controls, in normal people, they have a black band here, we call a band, a black streak, okay? So that's normal. So we can detect this enzyme. The enzyme is there. These are the patients with mutations, with 2 mutations, and they're nothing, right? You can see this. There is something here, there's nothing here. And these are parents, they carry just 1, and you see they have something, but less. So with this experiment, we showed very clearly that the SORD enzyme, these patients, they don't have it. These mutations, they destroy this enzyme, okay? So you have to live without it. And what is important, maybe here, the most important is this one here in this corner. Focus on this. In this experiment, we showed that in control, so in normal individuals without any SORD issues, the sorbitol, this stuff here that you don't want, is extremely low. In patients, in those patients that don't have something here, it's 100, even 500 times, sometimes higher, the sorbitol in their blood, okay? We have never seen anything like this. This is unbelievable. And these patients, they have this their lifelong. They have way too much sorbitol in their blood, and that damages their nerves over time. Now, how can we prove this more? To sort of shortcut things, we didn't develop a mouse model, which takes many years sometimes. We went into a fruit fly model, or Drosophila melanogaster. So in these flies, you can study this. They have the same gene, they have the same pathway. When you simulate this disease in these flies, I'm not going to go through all the details, but basically the nerves, they degenerate. So the fly eye is a little different in insects. As you know, insects have different eyes than humans. And you can actually see the nerves in the fly eye right on the surface. And you see there are these holes here. So clearly these nerves are not happy, they are dying. And as flies get older here, in this experiment, as they get older, 2 days, 20 days, 40 days, you see that the red dots, they're much lower, means these flies, they don't function as well. And in fact, what this measures here is how well they can walk. So they like to walk along surfaces, and we measure how well they can walk. As they get older, they don't walk as much anymore and as good than their peers that are normal. So that kind of showed that we can somewhat model the CMT phenotype, the weakness in these flies as they get older. And the most exciting piece is when we then took some of these aldose reductase inhibitors, and we treated these flies, here are the flies, and here are flies. Actually, this is the same experiment, how they walk. You see that, again, black and red, non-treated. As they get older, they don't walk as well. If we treat them, this is blue and green, they walk almost as good as the black ones. Okay, a little less good, but this is a big difference. I would say almost as good. So, and here we see the same in cells that CMT patients donated to us who have, these are from skin biopsies, these cells. So CMT patients with mutations in this gene, in SORD, you see that when we treat, so when we don't treat, as I showed before, the sorbitol levels are very high. When we start treating, they go back to normal, okay? Very strong evidence that this will actually work. And you will hear later today from a company, Applied Therapeutics, that has a novel type of these aldose reductase inhibitors. And they have already started some preliminary trials. I'm sure they will tell you about it. Great excitement that these drugs might soon come to CMT patients in the United States to be tested. We need to first test if they really work in patients, because humans are not flies, obviously. But we have great conviction that this will be a success story. So more on this later. Just to round this up, of course, this is further developing. Here are some key things we're working on right now. One is we are trying to identify more SORD cases. I'm sure you have seen the emails from CMTA. If you have an unclear CMT gene, so you don't know your gene, basically, and your parents never had CMT, you may actually, you may have the SORD gene, and we would be very happy to check on that. So you could contact CMTA, of course. You can also contact us in Miami directly, and we would then coordinate with you and see if you have the SORD gene. Because if you have, then of course you could potentially benefit from these upcoming treatment trials. We also, then what we do is we try to understand better what the CMT, what it looks like in patients with SORD mutations. We call this a natural history study. We have already identified close to 150 SORD patients now internationally. They come from, not just from the United States, they come from Europe, they come from Asia, they come from South America, from all places. You will hear more about this effort, about upcoming trials from Applied Therapeutics. And what we also do is to even better understand why sorbitol is such a bad molecule to have in your blood. We are in Miami, we are studying an animal model that is a rat. And in London, our colleagues study a mouse model. And we also in Miami study stem cells, which can then be made into nerve cells, motor neurons. And we study them in Miami in culture to better understand why sorbitol is so bad for nerves. So just this slide to show you that the CMTA has been a great supporter for our team in Miami here. We are currently receiving a quite sizable research grant to identify more genes for CMT and diagnose patients better that have not been diagnosed yet. And there are other grants that also are supported by CMTA. So with this, I just want to give you a quick acknowledgement to my team here. This is not just a Miami team, this is really a huge international effort. I think I convinced you of this. And I would be very happy to hear a few questions from you. Thank you.
Thank you, Dr. Zechner. We have one question I'm going to pull up on the stage, and then we'll have to conclude the session. But if any other questions come through, thank you. You see the question on the screen?
Oh, hold on. Let me switch over here. Yes. So somebody very educated, AT007 is the candidate drug, the new drug that is being tested, and you will hear more about it this afternoon. That's a very good question. Of course, the main regulator of glucose in your blood is insulin, right? Normally, the glucose, which is a very important sort of transporter of energy, is taken up into all cells really to keep things going. So in normal situation, the excess glucose would be taken up and regulated by insulin. That should not be a problem, I would say. But I want to say this, the more we learn about this entire pathway, there are certainly more questions to be answered and to be learned. So some of this, the details, physicians, scientists will have to go back and see what actually happens. So, but I would say I would fully expect that insulin will regulate this. Mm-hmm.
Thank you, Dr. Zechner. Thank you for such an informative presentation. As always, we always learn something new, and we greatly appreciate the contributions you and your team are making to gene discovery efforts in CMT. We are going to wrap up this session now and would encourage everybody to head over to the Type 2 Gene Therapy and Drug Development session, which is starting in about 2 minutes. Thank you again.
Thank you.